Today's speaker is Dr. Sarah Zanders, an associate investigator at the Stowers Institute for Medical Research. She has broad interest in understanding the effects of genetic conflicts by selfish genetic elements on eukaryotic genomes and their evolution. She discovered and has been studying a group of meiotic drive alleles in fission yeast. She has an interest in uncovering the general themes about meiotic drivers that could be useful in studying other natural drivers, as well as engineered gene drives to control natural populations. Sarah, thanks a lot for participating in our webinar series. We're really looking forward to hearing your talk. And at this point, I'm going to give the floor to you.
All right. Well, I'm really excited to participate in this webinar series. I've enjoyed it very much so far. So to try to get you in the mood for my talk today, I always start my talks with Skeletor here because I'm a fan of Skeletor and that he's a bad guy, and the genes I'm going to tell you about today are also bad guys. And so I think that this is really important to give some of our time as scientists to thinking about bad guys. And the reason for that is that bad guys are really important. So if we just think about our own lives, bad guys, or the threat of bad guys, shape the things that we do every day. They're a major part of our lives. So think about how many passwords you have, think about how many keys you use, think about how worried you are about your special stuff. Those are all kind of mundane aspects of life, but they're shaped by this threat of bad guys, even if we rarely encounter them face to face. And there's a similar situation that exists within genomes. So there are bad guys within genomes, including our own. These are kind of parasitic DNA sequences. And we're very interested in the lab of identifying these parasites and trying to figure out how they may shape everyday biology, just like the threat of bad people shapes the things that we do in our everyday lives. So the bad guys I'm going to tell you about today are killer meiotic drivers. And usually in my talks, I focus mostly on mechanism, generally talking to a bunch of molecular biologists, but I've really enjoyed the focus on evolutionary biology that's been a part of this webinar series. And so I wanted to talk a little bit more than I usually do today about the evolution of these things, because I think that that's particularly important. Specifically because I think the mechanisms of drivers are shaping up to not be super broadly conserved, but the evolutionary implications of these things, I think, is where the conservation is really going to be in this field. So I think the WTFs are great models to try to figure these things out. Okay, so let's start with defining the terms. So killer meiotic drivers, what do I mean by that? Well, quite simply, what we think these, or what we know these things do, is that they destroy the gametes that don't inherit them from a heterozygote. So they're stretches of DNA that are able to do this. So if we have a generic heterozygote here, where there's a little a and big A allele at this given locus, after meiosis, we anticipate that those two alleles should segregate into separate gametes. I'm showing sperm here just because they're the most fun to draw. But what could happen if one of these things is a meiotic driver is it can somehow destroy the gametes that don't inherit it. So in this case, little a is the meiotic driver. So this is really important on many levels. One is that this kind of activity can and does cause infertility in natural populations. It causes infertility when humans are trying to do controlled breeding, such as in crop breeding. So that's quite important for us from a human health standpoint. But also, these things are major drivers of genome evolution. So if we're interested in genomes and how they change over time, it's really important to acknowledge the existence of these parasites and try to figure out how they are affecting genome evolution. So if you've been participating in the webinar so far, you can appreciate that meiotic drive is quite widespread in eukaryotes. It's not found in just one type, but the mechanisms of drive are also quite diverged. So they're not all meiotic drivers are killer meiotic drivers. But I'm quite interested in trying to find general principles of drive that will hopefully apply across many systems of both natural and potentially engineered drive systems as well. And so we need a good model system to do that. And our favorite model system is probably the easiest to study in that it's a small single-celled eukaryote. Our base system that we study is the model fission yeast Pombe. And so this is a picture of the commonly studied lab isolate of Pombe. So Pombe is really great for studying meiotic drive and just meiosis in general because it has a very simple sex life. Usually Pombe cells are haploid. So each of these two cells, for example, is haploid. It has one copy of this A locus, and it's got this big A allele. But we can mate two haploids to generate diploids with two copies of this locus, so diploid, just like you and me. But then after meiosis, which we can easily induce in the lab, we regenerate haploid meiotic products or gametes, but in fungi we call them spores. So here we have 4 healthy haploid spores. But we can't study meiotic drive in this situation because we don't have any heterozygosity. Remember, you need heterozygosity, you need competition between alleles to see meiotic drive. And so one way that we can introduce heterozygosity into this system is we can mate the lab isolate here to a wild isolate. And in that case, now we would have a heterozygote, which, at the A locus and many others, you have heterozygosity. And as a postdoc, I did this in Hermite Moloch's lab, and this is what I found. I generated these heterozygotes, put them through meiosis, and rather than generating 4 happy haploid spores, most of the progeny of this process, or the spores generated, were dead. And when we could recover some and genotype them, what I found was that alleles, usually alleles from the wild isolate, were dramatically overrepresented in the offspring. And so we did a lot of work that I'm not going to describe to you now to demonstrate that both of these phenotypes, the destruction of spores and the overrepresentation of one allele type, those things were both caused by meiotic drive, and that there were actually lots of meiotic drivers acting that were present in both the wild isolate genome and the lab isolate genome. But at that time, we didn't know what any of the meiotic drivers were. We just thought they were there. So additional work that's published that I'm not going to explain, both in my lab and in Leland Du's lab, identified WTF genes as meiotic drivers. So the meiotic driver that we mapped was WTF4. So I'm going to talk a lot about WTF4 today, but just a preview or sneak preview of the rest of the talk, there are many WTF genes. So like the generic driver I introduced earlier, the WTF killer meiotic drivers destroy gametes that don't inherit them from a heterozygote. And these things cause infertility in fission yeast. And we believe they're a major factor driving the genome evolution of fission yeast. So I'll start now and I'll tell you a little bit about the WTF phenotypes that they cause and the mechanism, and then I'll go into the evolution. So here we are with phenotypes. So the first thing I want to point out is I'm not going to show you the phenotype of homozygotes because it's not interesting. Basically, nothing happens when there are 2 copies of a WTF gene at a given locus or 0 copies. What we see when we see a phenotype, it's specifically in heterozygotes. So I'm just going to show you that. So we can generate a heterozygote very easily by mating a haploid that lacks a given gene, in this case WTF4, to a haploid that has it. and now we have our heterozygous diploid. We can induce that to undergo meiosis, and what we see is this. We see 2 dead spores, and we see 2 viable spores. And we can genotype those to figure out which of the 2 possible alleles the viable spores inherit. And we find that in general, the viable spores inherit that WTF4 locus, and the ones that didn't are destroyed. I'll show you that quantified here. So this is the percent of the progeny or the spores generated by this heterozygote that inherit WTF4. And you can see it's almost all of them, whereas the Mendelian expectation is only 50%. So we get quite strong drive. Moreover, we can move that WTF4 locus around in the genome. With fission yeast, you can do pretty much whatever you want genetically. And so if we put that WTF4 gene someplace else, we find that it causes drive at that locus. And so basically all we need is WTF4 to cause drive at a given spot in the Pompe genome. So it's an autonomous driver. All right, so it took many years to map WTF4. So it was quite exciting for me to have mapped it, but it was also pretty confusing at first because I anticipated drive requiring more than one gene. And the reason I was anticipating more than one gene is because it was clear that in order to cause drive, the locus needed to be able to do 2 things that are quite different. The first thing is the driver had to be able to somehow distinguish between the spores that inherited the drive locus and those that didn't. So a self versus non-self recognition thing. And the second thing that the driver had to be able to do was to destroy those that didn't inherit it. So how does this one gene do these 2 quite different things? So the first 2 students in my lab, Nikki Knuckles and Maria Angelica Bravo Nunez, figured this out. So I'll tell you just in a cartoon summary what they figured out. So I'll just point out that much of the mechanistic stuff I'm going to tell you about today is going to be very cartoon based, but the work is published. And so if you want to see the actual experiments, they are available in publication form. Okay, so in this cartoon, we have our heterozygous diploid, where you have one allele of WTF4 and the alternate allele does not have WTF4. After meiosis, those 2 alleles will segregate into separate gametes. I'm just showing 2 of the 4 possible gametes here just for simplicity. And now, how does this driver work? Well, this is a more in-depth look at what the WTF4 gene actually looks like. It's got 6 exons. And what Nikki and Maria found was that the gene encodes 2 different proteins, and that's the key to the function of this placeholder. So the first thing that it encodes, the coding sequence is found on the last 5 exons, and it encodes a protein, and we call this protein the poison because we can see that it is a toxic protein. If one tags that protein with a fluorescent tag and looks at where it is in this sac or ascus that's holding these spores, what we see is that poison is everywhere. It's inside all the spores. It's surrounding all the spores. There is an overabundance of poison. All spores are poisoned. Now, if that was all that happened, everything would die and drive wouldn't work, but that's not it. There's a second protein encoded by this locus that has those same 5 exons as the poison, but it has an additional exon. This protein we named the antidote because even though it is structurally very similar to the poison, it has a different phenotype. It specifically rescues the toxicity of this poison, so hence the antidote name. And when we look at where tagged antidote protein is within asci, we find it only in those spores that inherit the WTF4 locus. So if we look at this entire picture, we can see that everybody's been poisoned, but only these spores that inherit the locus get rescued by the antidote, so these are destroyed. Just to give you a little bit of data to support that model, I'll show you this picture in which we have a heterozygous diploid in which we built separation of functional alleles that tag either the poison or the antidote with different colors. So we have the poison protein here specifically tagged with GFP. And if you look into this sac that's holding the products of a single meiosis, you can see that there is in fact poison in all 4 of the spores, and it's also surrounding all of the spores. Now, the antidote localization is quite different. Rather than it being everywhere, we see the antidote specifically in these 2 spores, and those are the 2 that inherited the locus encoding this. And so overall, the general gist of it is that this driver poisons all the spores and only rescues the spores that inherit it. So it's poison everybody, save yourself. All right, so we know a little bit more about the mechanism of this. And so this is the continued work of Nikki Knuckles, former graduate student in the lab. She wanted to have more a better mechanistic understanding of what made that what makes that poison protein toxic and how the antidote can counteract that toxicity. So again, this work is published. It's all described in this paper, but I'm going to give you a very cartoon overview of the conclusions of that work. So here I'm showing you the poison coding sequence. Again, it's these 5 exons. And what Nikki's work uncovered was that this protein is remarkably prone to assembly. We can't detect it, or we were unable to detect it in soluble form. The protein, we could only detect it in an assembled form. And so because of that, I like to think of the protein as a Lego. And so if we look within a cell, if it's expressing this poison protein, the different monomers of protein assemble into aggregates, and those aggregates are toxic. It is not entirely clear why those aggregates are toxic, but our best guess is that the aggregates disrupt protein homeostasis in the cell. So in less sophisticated terms, we know the proteins, excuse me, are are very hydrophobic. And so we anticipate that these proteins, these aggregates, may be bumping into other proteins, disrupting their folding, and non-specifically just kind of messing up the protein folding within the cell to the point that the cells can't manage that anymore and they die. So we're doing more work to further test this model. Okay, so how would the antidote counteract this toxicity? Well, the antidote, if you recall, has those same 5 exons that the poison has. So it must also encode this hyper assembly domain that we depict with the Lego, and it does. This protein also assembles in cells. We don't find we can't detect a soluble form. We always see it assembled. But the 2 proteins are not identical because the antidote has this extra domain here. And I like to depict that domain with this kind of sticky hand. And so how does this counteract that toxicity of these poison aggregates? Well, what we see in cells is when both proteins are present, they co-assemble into aggregates, which on its own would still be really bad for the cell. But this sticky hand domain has a very specific function. And what it does is it somehow alerts the cell's aggregate management systems that these aggregates are problematic and that they should be dealt with. So they kind of flag down the cell's aggregate management system. And the result of that is that these aggregates are actively transported into the vacuole of the cell. So the vacuole is like the fungal lysosome. So it's kind of like the trash can. So the antidote promotes trafficking of these nasty aggregates to the cellular trash can, And now these cells are fine. So there was no data there, but since we're used to looking at this picture, I'll just kind of illustrate some features of that model that are supported in this picture that you've already kind of learned to look at. So if we look again at this picture of the poison, I hope you can appreciate that we see that this poison tends to assemble into aggregates. And even this area where it looks like it's soluble protein, from other experiments, we've seen that even those are just smaller aggregates. And the antidote is also aggregated, but it's present in this kind of restricted region of the cell. And in other experiments, we've shown that that is the vacuole of these spores. And in the spores like this one that inherit the antidote, we can see that the poison is present in that same region, that vacuole region of the antidote. It's not distributed throughout the spore. Whereas these spores that do not inherit antidote, the poison aggregates are distributed all throughout, and that spore will be destroyed by those aggregates. All right, so I'll give you a second to reflect on mechanism, because we're going to make a transition here into talking about the evolution of WTF proteins. All right, so let's start with the expectation. When I started working on Drive, there is kind of an expectation of the evolutionary trajectory that a driver is going to take. And at the time I started studying Drive, pretty much all the drivers that we knew about supported this trajectory pretty well. So let me explain this expectation, and I'll do it in kind of pieces. So one expectation is that, a driver has to be born at some point. And when that happens, it gets this a transmission advantage through meiosis, and that can allow it to spread through a population. Now, unopposed by other factors, this driver could sweep to fixation in a population. And by fixation, I mean everybody in the population is going to carry this thing. Once that happens, then there's no longer any heterozygosity of that driver. And so it actually ceases to be a driver because it can't drive in the absence of heterozygosity. So in the absence of this driver being able to do anything, one would anticipate that it could decay or become extinct. So we've tested this kind of experimentally in the lab. So this is work of a graduate student in the lab, Ph.D. So what Ph.D. did was he used the features, the parameters that we knew about WTF4 to evolutionarily model what he thought the evolutionary trajectory, excuse me, of WTF4 would be if it was introduced into a naive population. So he did the modeling, but then he also tested his predictions experimentally and found very good agreement between the models that I'm going to show you and his experimental analysis. So I'll just show you the models. So what this is a WTF driver with the same features as WTF4, introduced it into a population at low frequency, and then we monitor how that frequency would change over time. So this is the time component, different generations. So Ph.D. did this under a number of different conditions. The first one I'll point out is if that population was mating randomly. And if you follow that gray line, basically what we see is the driver quickly spreads to fixation in a randomly mating population. All right, and the inbreeding, if that's happening in the population, which is something that we know does happen in POMBI, inbreeding will slow that spread to fixation, but it never stops it. So all of these lines eventually reach fixation too, it just takes longer. So as long as there is any degree of outcrossing, a WTF driver is expected to spread to fixation. in the absence of other factors. So basically, Fibo's work is quite consistent with this kind of initial prediction we had about the evolutionary trajectory of these things. But I tried to point out that drive to fixation was in the absence of other factors. And one other factor to consider is suppression. So meiotic drive is not good. It is not good for any number of reasons. I'll refer you to this review that Rob Unclis and I wrote a few years ago. But the gist of it is that natural selection works best when each allele gets a fair shot. And each allele gets a fair shot with Mendelian or fair allele transmission. And in the absence of that, or in the presence of drive, the same way to say that, there can be all sorts of problems that lead to low fitness or infertility. And those things can arise directly as a consequence of meiotic drive or indirectly. But basically, drive is bad. Natural selection is thought to favor suppressors of drive. So that leads to another branch of this expected evolutionary trajectory of drivers, is that it's quite possible that suppressors to drive will arise that can stop drive. And just like the top branch of this pathway, if a driver is suppressed effectively within a population, it no longer drives, it no longer gets a transmission advantage, and it's quite similar to the situation where the driver is fixed. If it's not driving, it can decay or eventually go extinct. So we have explored in the lab if this branch, this expected branch of the pathway, is also happening in WTF drivers. And so we did that by looking for suppressors of WTF drive. So this is work of Maria again, and this is looking at this driver called WTF13. Much like WTF4, this driver, when heterozygous, causes killing of the spores that don't inherit it. So you can see that quantified here. Just as before, this is the percent of the progeny that inherit WTF13 from this heterozygote. If everything was fair, we would expect 50%, but instead we see much higher than that, very strong drive. But this is an absence of the suppressor. What Maria found was that WTF13 had a suppressor present in some of the strains that we used in the lab. And in the presence of that suppressor, even if WTF13 was heterozygous, it showed no drive. So absolutely Mendelian allele transmission. So WTF drivers can be suppressed, and they can be absolutely suppressed. Has a fun twist. The actual identity of the suppressor of WTF13 was another WTF gene, an allele of WTF18. And so basically WTF18, rather than encoding both poison and an antidote protein, Maria found only encodes an antidote protein. And that antidote acts as a good suppressor of WTF13. So we've looked for other suppressors of WTF drivers. And so far, we've only found WTFs as suppressors of WTF drivers, nothing else. But still, nothing inconsistent with this initial prediction of how meiotic drivers should evolve. We see that they can quickly spread to fixation. We see that they can be suppressed. And so one would anticipate these things should be short-lived. So they should live hard, die young, and be restricted to one or a few lineages. And initially it looked to be the case. So the WTF genes were annotated to be specific to Pombi and not found in other yeast species. But we've explored this a little bit more carefully, and we did this first in parallel, again with Leland Du's lab, but then as collaborators. And so this work that I'm going to show you now is a fruitful collaboration with Leland Du's lab. And again, his lab is the one that also discovered that WTF genes cause meiotic drive in Pombi. Okay, so what did this collaboration find? Well, we looked a little harder at the fission yeast genomes. And as I've already told you, there are WTF genes in Pombi. But we looked quite hard to try to find them in Japonicus, and we failed to find any WTF genes in Japonicus. But we found that there actually are WTF genes in these 3 other species that weren't previously discovered because they are extremely diverged from the Pombi genes and from each other. So for those of you who are keen on bioinformatics, a standard BLAST search will not find WTF genes in these other species, but one of these iterative PSI-BLAST searches is able to find these WTF genes. So I've already told you that the multiple genes in Pombi can cause meiotic drive. And so the question was, are these WTF genes found in these other species also meiotic drivers? And the answer is yes. So we in the Do lab have functionally tested WTF genes from S. octosporus and verified that they are causing meiotic drive in that species. We haven't directly tested the WTF genes in Osmophilus or Cryophilus. These are non-model species that are not necessarily easy to work with. So we haven't verified that the WTF genes cause drive in these species, but we have functionally tested the proteins that they encode and found that in both of those species, the WTF genes encode poison and antidote proteins on overlapping transcripts, just like the Pombi genes I've told you about so far. So the conclusion of that analysis is we go back to the ancestor of those 4 species, which is here, which is estimated to have existed about 120 million years ago. And we can infer that this ancestor had WTF genes, and also that those WTF genes were likely acting as meiotic drivers, which means these WTFs have been causing meiotic drive for a very long time, much, much longer, over 10 times longer than we anticipated. So this seems to be wrong, or at least incomplete. So we've spent some time trying to investigate how is it that this model is wrong or incomplete, and can Pombi or these WTF genes help us figure this out? So again, this is wrong. What's the right answer? Well, what we think is that this isn't necessarily wrong, but that this is incomplete. So what we think is happening is that this pathway of evolution is not necessarily left to right, as is shown here, but that there are a number of pathways that lead backwards and lead to driver rebirth from any of these states. So I'll kind of go through in the next few minutes how we think this driver rebirth can be happening. So one mechanism that seems to be key to this process is gene duplication. You've already likely picked up on the fact that there's not one WTF gene in Pombi. I talked about WTF4 and WTF13. So let's look at this tree and I'll tell you just how many of these things there actually are. So here's Pombi. In the reference genome of Pombi, there are 25 WTF genes. And in some of the natural isolates that we study, there's even more. So up to 38 WTF genes in Pombi. In these other species, there's also many copies of the WTF genes, over 80, for example, in Octosporus. Now, all of these genes are not intact. Some of them are pseudogenes, but not all of them. So, for example, in Octosporus, there's over 50 WTF genes that appear to be totally intact. So there are a lot of these things. And so one factor, again, that's bringing about new driver birth is duplication of these genes. Now, the mechanism of this duplication, I think, is pretty important because this seems to be one of those shared themes amongst natural drivers. And so it should definitely be something one should consider if designing an artificial driver. Okay, so the take-home message here is that the duplication seems to rely on flanking repeat sequences. So if we look at POMBI, the locus, WTF loci in POMBI, they tend to have this organization where there is a transposon long terminal repeat, or LTR sequence here. There's about a KB of sequence, and then there's a WTF gene. And this is repeated over and over and over again. That's actually the origin of the funny name of these genes. The transposon here is called the TF transposon, and the genes were initially named for that association. So the W is with, and then TF for TF transposon. So how about in these other species? Well, initially we anticipated that the WTFs in those species would also be next to transposon sequences because we thought that that was a key to the mechanism of duplication, but we didn't see that. And in fact, Octosporus doesn't even have transposons. But instead, if we look within this clade of organisms here, we see that the WTF genes tend to be next to a different repeat sequence. And like transposons, these sequences are distributed throughout the genomes and found in many copies. And so these are sequences of the 5S rDNA. So these encode the part of the rDNA. It's about 125 base pairs. And what we tend to see most frequently is one of these genes on either side of the WTF gene. And then this unit repeated multiple times within the genome. There's another pattern that we see quite a bit in this clade of organisms, and that again, we have that same flanking 5S gene, but there's also another gene that seems to have been duplicated with the rest of this sequence. And we don't know what that gene is. We named it WAG for WTF associated gene. But we don't know yet what that gene does, if anything. All right. So we think this gene duplication, again, that's largely probably assisted by these flanking repeat systems or repeat sequences, and that gives us new drivers to be born. Another feature that we think promotes rebirth of these drivers is that they are highly mutable. So they exhibit very little functional constraint. So it doesn't seem particularly easy to disrupt the function with mutations, and I'll explain that in a second. But also there are a couple of regions within WTF proteins that seem to be kind of programmed genetically unstable regions. So let me kind of dive into this high mutability a little bit more in depth. Okay, so one of the features of these WTF genes within POMBI and then also between the species is their tremendous diversity. So I'll show you just to help you appreciate that diversity. I'm going to show you an alignment of the poison of WTF9 and WTF13. So these are two genes from POMBI. So this is divergence that we see within a given species. So this is an alignment. For those of you not used to looking at alignments, this is like really not a good alignment. So I'll just kind of walk you through it for a second. So if the two proteins share an amino acid at a given spot, you can see that the symbol for that amino acid is found here. And so as we go across the gene or across the amino acid sequence, you can see that there's very few places where these things share the same amino acid sequence. And you can also see regions like this where one of the proteins has a lot of sequence and the other protein doesn't have anything at all. So these things are crazy different. Just to orient you mechanistically with the way we think these things work, it's like WTF9 encodes a Lego that looks like this, and WTF35 encodes something totally different, although with a few similarities. So these things are crazy different, but what I find most striking about that is that even though they are so different, they tend to cause the same phenotype. So WTF9 and WTF35 both cause very strong drive. So even though their sequences are crazy different, they're still doing the same thing, which means that there's a lot of space mechanistically for the proteins to change their sequence, but still carry out the same function. They have very little constraint that we can see so far. Okay, so they can change dramatically without changing the function, but also they have a couple of regions in them that facilitates them changing dramatically at a DNA sequence level. So here is a logo of DNA sequence found in WTF genes in Pompe. So there's nothing remarkable about this sequence, except for that it is found in varying copy numbers in different WTF genes. And so if we look at this, so this repeat region is found in exon 3. Some WTFs do not have this region at all, whereas others have it in up to about 7 copies. This sequence it's in the coding sequence, so it does encode amino acids, so those are shown here. But what's quite remarkable about these is we can totally get rid of them. Or yes, for this one, some of these do not have even one complete copy of this repeat, but can still cause drive. So this repeat is present, it encodes amino acids, but those amino acids are not essential for drive. But if the repeat number changes, that can generate a novel driver with novel features. So even though this sequence isn't essential for function, it is important in that it can affect the function of the protein or the specificity of the protein. So I'll try to explain that again with the Legos, because that helps me a lot. So I showed you earlier that the antidote neutralizes the poison by assembling with it. And what we found through characterizing a number of these WTF drivers is that the antidote and the poison assemble because they have that same structure. They're encoded on overlapping protein sequence or overlapping coding sequence, and so they have the same amino acids. And so that leads to this homotypic assembly, just like one sees with Legos. But what we found is that if the copy number of the repeat section expands or contracts, that changes the structure of the protein such that this antidote with a different number of repeats can't assemble with that poison anymore. And so this antidote could no longer neutralize that poison. And so it can change the identity of a driver by changing the copy number of the repeats. And now that repeat region that I showed you on the last slide was in exon 3, but there's another similar region found in exon 6 of these WTF genes in Pompe. The other species also have a repeat region, that the one that's homologous to the exon 6 repeats of POMB. So this dispensable repeat region that gets bigger and smaller has also been conserved for over 100 million years. Okay, so we have gene duplication giving us new drivers. We have mutations that birth new drivers, because every time you change the sequence of the driver, the new mutant is no longer neutralized by that ancestral antidote, and so it gives us a novel driver, which again can lead to driver rebirth. But now we have one more thing that we think is contributing to this birth of new drivers, and that's ectopic gene conversion. So gene conversion doesn't get enough press. It's one of my favorite things ever, but I'll explain it in the context of the WTFs. So if we have a bunch of WTF duplicates, shown here A, B, and C, they have some differences between them that are reflected in these different colors. What can happen with gene conversion is that recombination events between these different genes can lead to shuffling of the information present there. And so that process can give us 3 essentially new genes. And because of, again, the function of the WTFs, this will give novel drivers with novel poisons and novel antidotes. Those novel drivers will not be suppressed, for example, by previous suppressors or antidotes, because they have to match specifically. So within this family, novel alleles are novel drivers. Okay, so these are all of these factors that are creating this very dynamic evolutionary landscape. And just to illustrate just how dynamic this is, I can talk a little bit more about diversity of WTF drivers within the POMB species. So within looking at different POMB isolates, what is the landscape of WTF genes? So this is work of Michael Eichbusch, a scientist in the lab. And so looking, first I should set the stage for this a little bit by saying there's not a whole lot of genetic diversity within the POMB species. Any 2 isolates of POMB are almost genetically identical. So there's on average 99.5% DNA sequence identity genome-wide. So not a whole lot of diversity genome-wide. But the case is quite different with the WTFs. There's actually 0 WTF genes that are fixed within the POMB, the POMB species. There's also 0 suppressors that are fixed within the POMB species. And instead, each individual isolate of POMB that we've looked at has a unique suite of WTF drivers and WTF suppressors. And that has major implications. So this is just what I just said. Every isolate has its own unique WTF genes. And so the consequences of that are really big. So if we consider 2 isolates, which otherwise are almost genetically identical, at any given WTF locus, because of this rapid evolution, they're going to have different sequences of their WTF genes. And so if these 2 isolates mate, they're going to generate heterozygosity. Those heterozygotes go through meiosis. 2 of the gametes will inherit one driver, 2 will inherit the other. And what ends up happening is that they destroy each other. Because again, the antidote of WTF-X can't neutralize poison Y, and vice versa, because they have different sequences. Because of that, within the species of POMB, there is reproductive isolation that's pretty strong even though the genomes are nearly identical outside of the WTFs. So that makes sexual reproduction not a lot of fun in Pombi if the organism reproduces by outcrossing. So these have a major effect on the breeding capability of Pombi. Okay, so I told you one of the reasons we study Pombi is that we're quite interested in general principles of drive, and I just want to highlight some of those here briefly. So I don't think that these features are going to be restricted to Pombi. Particularly this gene duplication has proven not to be specific to Pombi. There are other fungal drive systems, and as we've heard in previous weeks, there are the dox family of drive system in flies, and these things have the same kind of repeat mediated gene duplication of a given drive locus. So many of these other things kind of follow from, particularly the ectopic gene conversion kind of follows from the gene duplication. So I think that some of these things, potentially all of these things, will be repeated in other drivers, other drive systems. Okay, so those are the things I talked about. There are other things I didn't talk about for time restraints. This is another facet of the lab that is very focused on the effect that meiotic drivers may have on the evolution of meiosis. We've published one paper about this, and the gist of it is that we think that meiotic drivers have the power to make meiosis not work as well as we think it should. And this, again, we feel is a genetic principle that is mirrored in other drive systems, particularly in the work of Akira Takashi, who we heard talk in the first session of this webinar series. Okay, so with that, I will point out the folks who did a lot of this work. So this is a very old picture of our lab. We need to get a new lab picture, but it works because I talked about work that happened over many years. So I mentioned Nikki and Maria, so they've both gone on to postdocs now. They were graduate students in the lab. I mentioned Mike, he's still in the lab. And this is Mikael, so he was leading the collaboration with Leland Du's lab, looking at the long-term kind of ancient WTF present in the other species. Let's see, who else? Oh, I mentioned Phoebo's work, his experimental evolution work, and Blake was also, this is Blake, this is Blake, this is Phoebo. They both also were key to our evolutionary analysis. So with that, I will stop talking and answer any questions that folks may have.
Thank you, Sarah, very much for a very clear presentation and really fascinating group of meiotic drivers for sure. Lots of questions that come to my mind. Let me encourage the audience to either chat in your questions if you'd like, and some of you have already done that. That's good. We'll get to them in just a minute. Or you can raise your hand and you can just ask directly, and we're happy to unmute you as well. And Sarah, I'm just going to tell you that we're seeing your desktop at the moment, and you may not.
Oh, okay. I'll put this back up.
Yeah. Yeah. So let me, this. So wanted to ask you a little bit more about the nature of these proteins and what, if anything, you can say about them. And also anything you might be able to say about the biochemistry of these things and the interactions of these, of the toxins and the antidotes in terms of, is there a specific stoichiometry if you express these things in E. coli? Well, can you express these things in E. coli? Are they toxic in other cells? So could you say something about that if you know anything?
Yeah. So we have expressed them in other systems. So even easier for us than E. coli is budding yeast, another yeast species. And so a lot of what we know about how these proteins actually work, we've learned by studying them in budding yeast, so expressing them in budding yeast. So that's actually how Nikki kind of figured out this model. But essentially, if you express the poison in budding yeast, it will kill them. And if you co-express it with the antidote, it rescues. And that same dynamic that I told you about the proteins co-assembling and moving to the vacuole, that same thing happens in both Pombe and budding yeast. So biochemically, we don't know a whole lot about these proteins other than they really like to stick to each other. So they are predicted to have many transmembrane domains. So it could be that they are membrane associated, but we don't have a lot of evidence for that. It could also be they have predicted transmembrane domains because the proteins are very hydrophobic. And that could be what is allowing them to stick to each other so well. We are continuing to explore this now. So there's 2 graduate students in the lab who are currently trying to figure out this in more molecular detail.
Yeah, really interesting. Takeshi Akira has a question. It's actually a little bit similar, along the same lines at least. First of all, he says, great talk. He said, regarding WTF4, if you could express just the antidote and not the poison, are the cells fine?
Yes. So they are not totally fine. So I don't want to say that these things are. So let me see. So expressing just the antidote, in general, the cells survive, but you can tell that the cells would be happier without it. Let's say it that way. So both in Pombe meiosis, and if we express these things in budding yeast, we do see fitness consequences of expressing the antidote on its own. So it's not totally neutral. The cells would really rather not have it, but they survive. In budding yeast, the antidote expression will become toxic if we disrupt the cell's aggregate management system. So there's a whole bunch of stuff that the cells use to manage protein aggregates. And usually those things are not essential. So we can delete those factors and the cells will still survive. But if we delete those factors and express the antidote, then the antidote is no longer trafficked to the vacuole and the cells will die. So the antidote is also a nasty aggregate, but the cells are just better at dealing with it.
Okay, great. Hector Kamada has a question. It goes back to some of the evolutionary aspects of this, and just some clarification. I think trying to understand why or how the poison protein function would evolve and increase in frequency in the first place. Said I'd expect the protein to be so deleterious that the antidote wouldn't need to be born in response to the poison. So
let me go back to this slide. Let's see. So the key thing about this that I didn't explain very well, so I'm very thankful for the question, is when a new poison is born, a new antidote is simultaneously born. They are encoded on the same coding sequence. So this is WTF4, but this is the same for all the WTF genes. So if a mutation happens in this gene, it's going to change the coding sequence of the poison and generate a novel poison that would no longer be neutralized by the previous version's antidote. But simultaneously, because they use the same coding sequence, the antidote will be born at the same time. So it's very evolvable. So you can get a new poison at the same time you get a new antidote. And so that's part of the evolutionary freedom of these things. So for example, let's assume this guy, this driver exists, but it's suppressed. What can happen quite easily, because the thing's so mutable, is a change can occur that will generate a novel poison and its corresponding antidote, but that new driver will be immune to the previous driver's suppressors. So we think that that kind of thing, I don't know, makes these things very nimble in terms of evolution.
Yeah, great. Let me keep encouraging the audience to ask questions, and feel free to ask whatever you'd like. And because all these questions actually allow Sarah to talk more about the system, and we get more information for sure. You can also raise your hand too. Feel free to do that. Don't be shy. There was an aspect of this that you didn't mention, and that's really interesting, and that is you're getting two proteins from this transcript, but there's a timing issue here that seems pretty critical in terms of the poison needs to be expressed first, and then subsequently the antidote needs to be expressed at a particular time. So what is the nature of this regulation of these two proteins? Is this two promoters? Is this a translational control?
Yeah, we have a project looking at just that. So we have a paper in revision that's dealing with that. So that's I mentioned Nikki, but that's another graduate student is completing that, Ananya. And so what that project has shown is, yes, there are two promoters. So the promoter for the antidote is upstream of the gene, and the promoter for the poison is actually within this first intron. So they do have totally separate promoters. The antidote we've recently found is not only expressed in spores. So our initial tagged construct, the one that you can see in, let's see, this slide. So we initially looked at the antidote using this N-terminal tag, and this is what we saw. We saw nothing except for in the spores. Nikki and Maria, or not Nikki and Maria, Nikki and Ananya have subsequently characterized a different tag that reveals additional antidote protein. And so the most current version of what's happening is that very early in the process of meiosis, we see that there is antidote expressed. So there's antidote there before there's any poison, which is good for the cell because the poison's really nasty. After a bit, there's poison expressed. And so when these spores start to form, there's actually poison and antidote already present in that shared cytoplasm in which the spores form. But then when the spores form within that common cytoplasm, something really cool happens. So the antidote that's present in that pool is excluded from the newly developed spores. So the pre-standing antidote population never makes it into a new spore. Whereas the poison that is present, some of it does make it into the spores. So there's differential inclusion of poison and antidote into the spores. And so when the newly formed spores are born, they have poison in them, but no antidote. Then a new burst of antidote is made, which is what we can see represented with this tag, that then protects only those spores that inherit that locus. So it is quite a bit more dynamic than this initial picture shows. So we're working on that. That should be published soon, I hope.
Yeah, fantastic. Pratima Trinuri has a question. She says, thank you for a very interesting talk and clear talk. Evolutionarily speaking, is it possible that these bad guys initially emerged as good guys? For example, a toxin emerges from a duplicated mutated gene, then our selfish drive emerges to counter it in one strain. Hopefully you're reading this along with me, so you not have to try to follow my reading of this. However, when this strain meets another, the selfish hero drive is now the bad guy. Perhaps a case of if heroes live long enough, they become villains. So
yes. Yeah, no, I think that's quite possible. The challenge with the WTF system, though, is that it is so old. So because these things have been around and doing this nasty thing for 120 million years, it's really hard to figure out what they might have been doing before that, even in the absence of all this extra rapid evolution that they're doing. We are very much interested in how one of these things could arise in the first place. But this is one situation, one question, which I think the WTFs are not great models to answer because they are so old. But because they are so old, to get to the last part of your thing about, well, I guess it's the opposite of what you say. So you say if heroes live long enough, they become villains. That's possible, but it's also possible that if villains live long enough, they become good guys. So it is possible that because these things have been around for so long and there are so many of them, that there could be things that they are doing for Pombi that are good that we don't appreciate. I don't think that's the reason they exist or the reason they've been maintained. I think they exist and are maintained because they cause meiotic drive, and that's incredibly beneficial to them. But in Pombi, for example, there are four WTF genes that are related to the others, but they don't cause drive. We don't know what they do. We can delete them, so they're not essential, but it is quite possible that they've been domesticated in some way and are doing something good for the cell. We haven't found that, but it is a possibility.
Great, thanks. Eric Lai has a question, and it's touching on a subject that I was hoping to get to as well. And anyway, he says the complexity is beautiful. I was wondering, in general, can you design synthetic drivers based on other aggregating domains? Or do you think there is something very specific about what FTF proteins do? Are drive conditions very sensitive to chaperone or other quality control mutants?
Yeah, so I think building a synthetic driver based on these aggregating domains would be a good idea. And I can't think of anything better. I mean, I have limited biochemical knowledge, but the WTFs are remarkably good at this. And so one of the things that we're kind of interested in doing is figuring out what a minimal aggregating domain to do this kind of effectively is. But such a thing would be quite good for building a synthetic driver. I mentioned that we test these things in budding yeast, but we also know that WTF poisons will kill animal cells as well. So aggregate management systems are quite well conserved in eukaryotes. So just basically building WTFs and other systems should build a good drive system if they're adequately expressed. So let me see, what else was the question?
Yeah,
I don't think there's anything evolutionarily specific about what these things do that's specific to fungi or Pombi. We have tried to suppress WTF proteins or the toxicity of like the poison by overexpressing chaperones, and it doesn't work. So the problems caused by WTF poison in general are not easy for the cell to deal with. We've done rather extensive screens in budding yeast looking for mutants that are able to overcome the toxicity of the WTF poison, and we haven't found any. And the genetic power of that system and my confidence in the Anunya's work make me think that it's very hard for cells to suppress this. And that's also consistent with the long-term evolutionary success of these things. It's hard to get around having a cell full of nasty aggregates.
Yeah. So Eric's clarifying a bit. He says, if you have a chaperone loss of function, for example, would the antidote not work?
If you have a chaperone loss of function. So haven't found any chaperone loss of functions that make the antidote not work. What we found that makes the antidote not work are the pathways that recognize and traffic aggregates. So those don't necessarily rely on chaperones. So there are pathways that recognize nasty misfolded proteins in cells, and those then can traffic them to, in our case, the vacuole or the lysosome or whatever kind of cell you're dealing with has.
Yeah.
But we've done a screen, for example, of the budding yeast deletion collection, so deleting all the non-essential genes and looking for mutants in which the antidote becomes toxic. So these are mutants in which the cells can't deal with aggregates. So we've published that list of things if someone's particularly interested in the factors that are necessary for the antidote to carry out its molecular function.
Yeah, great. You mentioned heterologous expression in animal cells, which was also toxic. Have you explored that heterologous expression beyond that? These I take were probably tissue culture cells. And I'm thinking like, could you make a fly system that is that's using this as a driver?
Yeah, we haven't done much to develop this thing as an artificial driver. So I'm at the Stowers Institute, which is kind of known for its love of non-model systems. So the animal cells that we've actually tested this in are nematostella cells, which are sea anemones. So if you encourage sea anemone embryos to express WTF poison, they die. But we didn't, carry out that project to its natural completion. We just kind of stopped it at the point where the sea anemones die if they express the poison.
Yeah. Scott Cheney has a question. If different Pombe isolates are reproductively so isolated, why is there so little sequence difference between them?
It's time. They haven't had enough time to accumulate a lot of differences. So the isolates that have been sequenced are estimated to have shared an ancestor about 3,000 years ago. So that's not very long. So genome-wide, there haven't been a lot of changes that have accumulated in such a small period of time. Excuse me. There are other regions of the genome that are much more rapidly evolving, like the WTFs, and like some other repetitive regions of the genomes, like centromeres. Those have changed more because they change faster. But in general, the different isolates are quite similar to each other just because they shared an ancestor yesterday.
Yeah. Great. Well, Takeshi has another question. It says, if I'm right, I think it's quite rare to have this many introns for fission yeast gene. Understand that the first intron is a promoter of the poison, but do you know if the other introns are important for drive?
Yeah, that is like I told you that like gene conversion is like my favorite thing ever. So
just say you have your screen up again, your screen sharing.
Okay, there's nothing I'm embarrassed about on the
Okay, just letting you know.
I don't if there is I can stomach it, but I want to find this gene conversion slide because that's I think the answer to Takashi's question. So this gene conversion thing doesn't show you the introns. So this is assuming that there's no introns here. But the intron exon structure is another feature of the WTFs that's been conserved over 100 million years. So we know that we can get rid of the introns and still have functional drive. but yet the intron structure has been maintained for like as long as these things have been around. And so, as you mentioned, the first intron is quite important. It's the promoter for the poison, but why maintain all those other introns if they're not functionally important? So I think the answer might be gene conversion. So this is my hand waving answer that maybe we'll model at some point to see if it's can be supported. But I've hopefully convinced you that these things can persist because they evolve very dynamically. They need to change frequently. And one of the things that gene conversion can do is this variant shuffling that I'm showing you here. Then that is good for the WTFs. That helps them diversify. But another thing that gene conversion can do that is not good for diversifying is it can homogenize. So homogenization would be really bad for the WTFs because then there's basically one antidote could suppress all the poisons if they all had the same sequence. So I think the introns buffer against homogenization because gene conversion track lengths tend to be kind of fixed, not totally fixed, but of a given size on average. And so if you expand the size of the gene, a smaller fraction of the gene will be gene converted in any gene conversion event. And that will kind of promote variant shuffling while also kind of limiting homogenization. And so the WTFs can then benefit from this shuffling while hopefully avoiding the homogenization, which could lead to their demise. So that's my guess for why the intron structure has preserved for so long. It's almost like the gene structure of these things is better conserved than their amino acid sequence.
Go back to Scott. Scott Keeney is full of a follow-up that I think you can see there. He says, thanks, the time argument makes sense, but wouldn't you expect to be able to find isolates that share the last common ancestor much further back?
So there have been a lot of isolates sequenced so far, and none have filled out the tree any better. So of all the isolates that we have sequenced, their ancestors appears to have existed 3,000 years ago. The next branch out is these other species. Let's see, let me find a slide that has a tree on it. The next branch out is that, this clade that has all of these species. So we haven't found other things that could help us get a more refined evolutionary picture. These things are all quite different from each other. And the only branch that we have sequences of many isolates is this Pombi branch. And all of those isolates that we have are very closely related. So we would have a lot more power to answer more fine scale evolutionary questions if we had more isolates, but right now we just don't. So hopefully as folks continue to sample new places to find new yeasts, we will be able to fill this out. And that's proved true in the last few years. For example, Osmophilus here was just found in the last few years. And Blake Billmeyer, the postdoc in the lab, and Mikhail actually assembled the Osmophilus genome in order to find the WTF. So this one was just recently discovered. So hopefully it was found in like beehives or something. So hopefully more sampling and other maybe insect associated habitats will yield more fission yeasts and will allow us to answer more fine scale evolutionary questions in this clade.
Great. Fatima asks one of the last questions. I think we're getting a little close to time, but is the variant shuffling a result of unequal recombination, that is intra or interchromosomal, or perhaps both?
So the kind of recombination that's happening here appears to not be the kind that we hear about most frequently, which is crossing over, which is exchanging of chromosome arms. Instead, it looks like it's gene conversion without associated crossover. So it's just as if a double strand break, for example, in WTFA here, used sequence from WTFB to fix. So a little bit of B would be copied to A in that case, but there'd be no exchange of chromosome arms. And so it is, it can be within the same chromosome, and it can be between different chromosomes. So these genes are distributed throughout the genomes, and they all seem to be participating in this gene conversion.
Yep, great. I think with that, we'll call that the last question. I want to thank the audience for the questions and the discussion. Thank you, Sarah, for hanging in there and continuing to answer the questions. It's a great sign that people are asking questions, as you know. They were listening and were really thinking, so it was really great. And I just repeat that again, that I thought your presentation was wonderful, and we're really happy that you participated in the webinar program here for us. I want to say that while this is the last webinar in this series, as I mentioned in my introductory remarks, we will be continuing, we're starting a new series in 2 weeks' time from today. It will be covering a slightly different topic. It will actually be looking at demystifying the Convention on Biological Diversity. So we're going to go into some more governance-related topics in terms of these genetic control programs. So with that, Sarah, I want to thank you again.
Thank you, everybody.
Yep, and thank you to the audience for all your questions. And we hope to see you back in 2 weeks and on a slightly different topic. Have a good day.